Well, welcome, Sandosh. Welcome to myDNA, and thanks for taking some time out whilst you're in Australia to talk with us. Over the last 4 years, I know you've been pioneering this world of personalised medicine and really helping taking personalised medicine from research and into clinical practice. And we're very excited at myDNA to be a partner with NHS Scotland to see this project come to life. And you've very much been, as the chief investigator, the driver behind it. And my understanding is it's one of the largest pharmacogenomic trials happening in the world at the moment, in one of the biggest hospitals in Europe. So it's very, very exciting to talk to you about preventative health, personalised medicine today. But before we start, maybe if you could just provide a bit of background on how you got into this, on yourself, and how you got into this field in the first place.
Thank you, Allan, and it's been great to be here. So, I'm a physician, and as a doctor, I've been seeing the same problem again and again. We give the patient the same drug, same dose. You see one patient, the drug responds well. Another patient, it does nothing. And the third patient, it can actually cause harm. And it's been extremely difficult to try and find a way to reduce the trial and error period of getting a patient on the right drug. A large proportion of this difference in drug response is due to pharmacogenetic. It's due to variations in the patient's DNA. So the science is not new. We've known that for a long time. The word pharmacogenetics was coined in the 1950s, but we are still nowhere near implementing pharmacogenetics at scale. How I came into this. So my background has been, in addition, I'm a clinical academic, so my research has been in large-scale population genetics. We formed a team in the early 2000s when genome-wide association studies came through. We did one of the biggest and the most successful genome-wide study for hypertension. And we discovered a gene for hypertension, a new gene, a new pathway for hypertension. That discovery phase took 2 years. But after that, connecting the SNP to a gene, doing the functional studies and doing a clinical trial took 15 years.
Yeah.
And after completing the trial and publishing it, we are still nowhere near trying to get that adopted. So that's a problem. And it's not unique. And so I'm coming from the discovery side, it suddenly exposed me to the problem. And pharmacogenomics is the problem where the gap between how much we know and how little is deployed is the widest, but the most fixable. And it's around that time that I thought that we need to solve this implementation problem. And fortuitously, at that time in Glasgow, we got substantial funding for a concept called the living laboratory. The start of that aligned with the publication of our trial in hypertension. And so it sort of smoothly moved into I had an opportunity to scale up the implementation aspect and bring the understanding, knowledge, and the experience from my discovery research into implementation science.
Fascinating. I know when we first started the company, and I started with my father, which was also roughly about 15 years ago. My background was not in healthcare, and my father being a clinical geneticist, him saying we need to make this genetic testing more accessible to people. I thought it would take 5 years or 15 years on. I agree, we're still solving that problem of how we get this into implementation. And I think when we first heard about the living laboratory and what the problem you were trying to solve, both me and myDNA and the business thought this is exactly why we started the company. And so we formed this partnership, which is really exciting. And you did cover it, but maybe just for those people that are not as familiar with the pharmacogenomics term, could you just explain what that means in simple terms again?
Yep. So pharmacogenomics in simple terms means if you take 10 people with the same condition and give them the same drug at the same dose, some of them will respond, some of them the drug does nothing, and for some it may cause harm. This what we call inter-individual variation in drug response. Majority of that variability is written in your DNA. And your DNA, as you know, is just a sequence of letters. So these could be changes in either a single letter in your DNA sequence or a segment of your DNA sequence. And that has an impact on how your body absorbs the drug or modifies the drug, activates the drug, or clears the drug from the body. And that has the implications on side effects or efficacy. If this science has been known for a long time, if you look back historically, as old as 2,500 years ago, Pythagoras warned about eating fava beans, and that is linked to a pharmacogene called G6PD. So pharmacogenomics has been known for a long time. If you look at the discovery of pharmacogenes, that's been progressing even before we got the human genome sequence. So the science is very, very mature. The most interesting and the most useful bit about pharmacogenetics is this is a sequence change in your DNA that can predict your response to drug or an individual's response to drug throughout their life. You test once, the test stays with you throughout your life. That makes it a really useful test both for the patient and for the hospital, the healthcare system. The complexity for pharmacogenetics has been trying to get this into clinical practice. That's a problem. The science is very mature. So if you look at our regulators, the FDA, there are about 600 drugs with a pharmacogenetic label, indicating that all these drugs could have some information sitting in your DNA that indicates how you would respond to it.
It's quite amazing, really, when the science has been around for so long. And often when I talk to people about personalized medicine or pharmacogenomics, they say, well, why wouldn't everyone have this information? Or why doesn't my doctor have this information on me? And you often hear about people's stories about them taking a medication and having side effects. And if they could avoid that by a simple genetic test, it just makes sense. And this implementation gap is really, the area that you've been focusing on. And so if we talk about how do we scale it and what the strategy is to get it into healthcare, I know Genomics England are doing a lot in terms of they've put together a 10-year plan that genomics is pretty much the cornerstone of that strategy. Can you tell us a little bit more about what their plan is and how they plan to use genomics?
So the NHS 10-year plan, NHS England 10-year plan, for the first time has included or placed genomics as the center of their strategy. This is the first time ever they put that on paper. And genomics is part of the 5 pillars for the healthcare service. Genomics is one of them.
Yep.
What's important to understand about that strategy is the ambition. The ambition is to build a population genomic service. And that is new. The goal is in 10 years, they want at least 50% of all patient interactions to be informed by genomics.
Wow.
So by 2030, that's their goal. NHS England, they already have their genomic service. They're going to add this population genomic service on top of them. And pharmacogenomics and polygenic risk scores are the main tools they're going to implement. So it's quite ambitious. And what's important about this, and the reason why genomics has come about, again, I'm saying the science is not new. But the important bit and the learning there is why it's likely now that we will see implementation of genomics happening at scale is because of 3 things. One is you have the science which has become very mature. The second is the cost of genotyping has collapsed. And thirdly, for the first time, you have full alignment between the policymakers, the healthcare leaders, and the patients.
Yes. Yeah.
And the delivery infrastructure.
Yep.
They're all on the same page.
Yep.
So you see convergence on that. Without that last bit, this wouldn't have happened. So now you're seeing a real change amongst the decision makers that genetics is something that can scale up and be delivered effectively.
No, it's very exciting. I mean, if you look outside of healthcare, you still think 10 years is a long time to bring that research or that innovation into healthcare. But seeing it to be such a strategic pillar of the strategy is very exciting for all of us genetic enthusiasts.
Absolutely, yeah.
And so I suppose the question is, in implementing this type of testing into a hospital setting or mainly let's talk about primary care in the hospital setting, what are some of the major barriers?
We have solved a lot of issues, but there are still a lot more aspects that needs to be solved. So the first thing, if you look at acute healthcare service, the first important bit is speed. It is the turnaround time. How quickly can you get a result to the physician? In a hospital setting, you need the result before the drug is prescribed or before the patient gets discharged. The average hospital stay for a patient in Scotland is about 4 days, 4.5 days.
It's a quick turnaround.
You need to get the result then. So that needs to be sorted out. The second and most important bit is the last mile. The last mile is getting the results from the lab integrated into the prescribing system, along with decision support that can convert those into understandable, easy to use recommendations to the physician. That last mile is it's infrastructure technology solution. It's not science, but that's the critical bit. So that needs to be developed. The third one is education. We are expecting thousands of prescribers to suddenly start trusting and acting on a test result, which they have not been trained in.
Yeah.
So we need these 3 pillars to be sorted out. And what you understand here is there is no discovery of science. This is all infrastructure and delivery.
Infrastructure and delivery. No, it's fascinating because I know a lot of doctors that perhaps even when you went to medical school, pharmacogenomics was not necessarily on the curriculum. It's something that's happened through your career, and now it's how do you change management and behavior change to get doctors confident to act on the recommendations of a genetic test. So I appreciate there's a lot of work to be done there. And I think that leads nicely onto the Phoenix trial, which is, something that you've been spearheading, which is a partnership with myDNA, and it's looking to engage about 4,000 patients with pharmacogenomic testing and see how this can be implemented in a real world setting. Can you tell us a little bit about what the goals of the study are and how that's going to help drive this last mile or this adoption that you speak of?
Phoenix has an origin story, and I think that's important.
I like a good story. Yeah.
So I mentioned the living lab.
Yes.
So the origin of Phoenix started about 5 years ago when we, from the university, so with Dame Anna and us, we proposed to the UK government, who were interested in funding. So they had this funding called Strengthen Places. And the Strengthen Places Fund is about economic growth, regional development, and inward investment. It's not medical. It's not
a
science.
Medical
No.
fund. Yes.
That was the call, and this was given to the region. So from the university, we proposed that accelerating implementation and adoption of precision medicine in innovations is the crucial bit. If we can solve that, then not only will we see impact on patient health, but more importantly, this is going to be an attractor of inward investment, innovations, and regional economic growth. So that was our proposal. We submitted to the government and we got the funding. The flagship work package was pharmacogenetics, which I led and lead. So we got the funding, and then came the opportunity for us to look at how to solve this. Because this was not a science grant, all our efforts were focused on how to solve the implementation question. The advantage we had because of the living lab, which got £37 million funding to actually build the whole setup for implementation. We had that safety net to really think agnostically, really build something from ground up. I went about looking into this. We got leaders from the industry, from government, from the NHS, all giving their opinions on what the problems were. So we worked, it took a couple of years, we worked around this, and it was very clear that a lot of research has happened in pharmacogenomics, but nobody has produced credible, compelling evidence that was palatable to the decision makers and the payers. And
Yes.
ultimately it boils down to credible evidence. And that means doing a clinical trial. And we knew that everybody who tried clinical trials did try, but they were all at a smaller scale. If you want to implement pharmacogenomics, if you want to change views of decision makers, you need to get the evidence. And you need to get the evidence that they would like to see. It's about efficacy, it's about cost effectiveness. Then we hit another challenge, the PREPARE trial. The PREPARE trial was ongoing in Europe at that point, and that was a big European grant, 7 countries, nearly 50 million euros. And they were almost halfway through the trial. So we got pushback from various people saying that the PREPARE trial is already ongoing. Why do you want to do a trial? Why do you even want to do pharmacogenomics? Just wait for the trial to come through. We looked into the PREPARE trial, and there were lots of, they were doing a big trial, and it's actually good for us because the fact that they did such a large trial meant it de-risked us running a trial. And we could do a trial that builds upon and adds to, extends what they were doing. They were looking into one aspect of pharmacogenomics, but it was not the complete story. And pharmacogenomics is a complex field to do trials in. So they did one part. We knew that there was a huge gap still to be filled. And that's where we decided to build our program. And again, we focused on trials. We had a lot of pushback from various industries. We're talking about, real multinational conglomerates coming and telling us that there's no point doing a trial. Pharmacogenomics is all done. You just do a small study and get it adopted.
It's not done, is it? Because it's not being implemented.
It's not
being implemented in hospital, and it's not standard of care,
Absolutely.
which I think is where,
It's not going to work, because the problem with the trial is the unknown. You're doing a trial. You don't know the answer. It may not be in your favor. But we knew that this would work, and it became more and more clearer that we need to focus on the trial. And this is where I think it's crucial to understand. Because of the living lab, because of the university, we were able to stick to the evidence and to what needs to be the tough questions.
Yes.
And not dilute it. So we did that, and there was a lot of failures on the way. We learned quite a lot. And time was running out. Living Lab had a strict 5-year funding window, and this took about 3 years now. And that's when myDNA came in. And what we needed was a company with the foresight that, this trial is a crucial stepping to understand that. There's not a commercial. There is commercial aspects involved, but to reach there, the goal, you need to do this trial. So myDNA, so less yourself coming in, took the risk and said, let's go for it. So that's one. You had the foresight and you trusted us to do the trial. From our NHS colleagues, they trusted us because we do lots of trials. And when I proposed to them that, this is a trial, we need to recruit up to 4,000 patients, they said, it's ridiculous. You will recruit only less than 1,000 patients in 3 years. And that's getting 1,000 patients is a stretch. You will not cross that. But I had done the numbers. This whole development phase, I had worked out everything, including the numbers. I worked out the footfall in the patient in the hospital and how many patients would be prescribed the drug. But they had a legitimate concern that this trial will not succeed because all the trials ever done could not get the rate of recruitment as required for Phoenix. So they said you will never get. But again,
and a lot of trials fall down because they can't
absolutely
recruit patients. Yeah.
Absolutely. So that's where this comes in, because I had completed the other complicated trial for my hypertension gene. It was a small trial. So compared to Phoenix, we had 240 patients.
Yes.
But we had to screen 1,000 to get 240. So, but only 240 had the intervention. So Phoenix was, by design, a complex, one of the largest trials we designed, and one of the largest trials to be done in such a short period of time. But it took about 4 years to come to that stage. So, and it was 4 years of concentrated work. So when myDNA came in, it was on the background of all this, but still there was risk. But I had ensured that everything was done, and when I explained to the NHS, they agreed that, you know, the numbers look okay, but they didn't believe that we'll get the numbers. So that's how finally Phoenix started. Lots of delay, and our problem was Living Lab has a fixed end date, and all this delay reduced the period of time that we had for recruitment. So we have less than 2 years for recruitment. We have now, in 14 months, as of yesterday, reached 2,470 patients randomized. This is, to give you the context about why we say this is one of the biggest and the most successfully recruiting trials, is it's not a real comparison, but it's a good comparator for the scale of what we are doing. During COVID, at the height of COVID, you had the whole country, the whole NHS, everybody working on COVID, trying to find solutions. And at that time, there was no treatment. Then the Oxford group came with this recovery trial. And that recovery trial identified the first ever treatment that can reduce mortality in COVID, and that was dexamethasone. And that was done as a trial across the country. The entire NHS across the country were working to recruit patients. Our recruitment rate is comparable to that from one side. So that's the scale. And the most important bit is the implication. What does this number tell us? It's not about the rate of recruitment. The number 2,470 in 14 months means it tells us how pervasive drugs with pharmacogenomic implications are being prescribed in the hospital.
Yes, of course.
It tells us that, you know, it's a huge need, because everybody who comes to hospital is getting a drug prescribed which has a pharmacogenomic implication. And we were able to recruit them. We also know that we've recruited over 26 specialties.
Amazing.
So it's not fixed to one specialty. It's the whole hospital. It's totally democratic.
Yes.
And it's good to see this. The design phase, sticking to doing a trial, taking the risk, and then showing that actually pharmacogenomics is such a huge problem that can be solved with what we have, and we can solve it.
Yeah, absolutely.
So it's been quite heartening to see that. So we will reach 3,000 patients by end of September. That's when the Living Lab funding, and then that's a good enough number for it. We aim to do 4,000. Yeah, but I think we will cross 3,000.
It's a fantastic outcome, and you know, I think your persistence over this period and passion to stick to, I guess, the plan of solving the problem, you know, the problem of implementation, I think is now a real fantastic sort of exemplar project to show that we can get the data and the evidence to get this type of testing into the NHS.
Absolutely. And the other important thing is, during this development phase, I have discussed with the government. I went to the genomics group in the government and all that, and they told me, you know, do not show your face to us unless you tell us how much money
you can save.
Pharmacogenomics can save. Yeah, they were pretty brutal.
We offer pharmacogenomics in the US, in Australia, and the UK, and we often get asked, do you have any health economic data that you can share to show the benefit, to go to that next question on widespread adoption and scalability. So I think this data is crucial, and you were definitely one of the drivers of sticking to the plan and not deviating from producing that evidence. And so congratulate you on all your hard work and getting to that point. And I think once we have this data, I suppose the next question is, if we sort of look at the future vision of personalised medicine, and if we think 5-10 years ahead, how do you see this all playing out?
I think Phoenix will be a major. We'll see what the results are, but we know how the results will go because we know the science.
Science is solid. Yeah.
Phoenix built on Prepare, scaled it up, and actually asked the much harder questions. And we are doing health economics as an a priori outcome of the trial. Then there would be other studies coming after Phoenix. End result will be what most of us keep telling is pharmacogenomics or genomics in general is infrastructure. It's not a special test. What we want is in 5 years' time, when pharmacogenomics is fully adopted, we will disappear. Nobody will talk about us because it's infrastructure.
Yes.
The genomic, a person's sequence will sit quietly, silently in the health record, happily waiting for the day when somebody prescribes, then it triggers a decision, a recommendation. The physician prepares a tailored treatment for the patient. So that's what I think I mentioned. That's called boredom. It becomes genomics becomes boring. It's like a standard accepted practice that you completely forget about it. But more importantly, if you look at it from 3 angles, from the patient perspective, patients recognize and they understand that the treatment they're being given is using their genetic information, is tailored to them, is personalized for them. So they will have the confidence that they're getting the treatment. It's not trial and error. Doctors, all their frustration with trial and error medicine is out because now they have at least a proportion of some of the difficult decisions being supported by pharmacogenomics, and they know that patient safety is one of their priorities, and now they have something that they can fall back on. And then you've looked from the perspective of policymakers, healthcare providers. For them, it's about improving outcomes and saving money.
Yeah.
So from the 3 perspectives, if just integrating genomics into regular clinical pathways will be transformative. And this is reflective of what Genomics England is trying to do over the next 10 years. And it's the same thing, getting genomics into healthcare infrastructure.
Well, it's very exciting, and congratulations on how the project's going so far. And you've been a true pioneer in leading the transformation or this revolution of getting genomics into the medical record and making it standard of care. So thank you very much for your time.
And thank you for everything you've done on the project. Thank you.